Ebola and its cousin Marburg virus have been with us for decades. They are largely a disease of the African continent. The last major outbreak was back in 1976. The current outbreak is about 20 times greater than any other combined infectious disease outbreak ever. Before now, many of the outbreaks were small, and they occurred in isolated regions in Central Africa, particularly the Democratic Republic of the Congo. Low-density populations with minimal travel kept the disease isolated to these small villages. The current outbreak is located in the more populated West Africa, in a time where transportation is more readily available. To date, there is no suggestion at this time that the virus has mutated and contributed to the outbreak. The bulk of the cases begin in Guinea and spread to Sierra Leone and Liberia. Ebola is a type of filovirus. Its transmission occurs through the ingestion, inhalation, droplets, unprotected mucous membranes, blood, bodily fluids, which include feces, vomit, urine, semen, breast milk, saliva, and tears. And, of course, through broken skin. There is evidence that the virus might also replicate within the sweat glands. Although the virus replicates within the respiratory secretions, we're not sure if it can be aerosolized. Transmission via the respiratory route, which include coughing and sneezing, is rare, if it happens at all. Airborne transmission has only been reported among healthcare workers exposed to aerosols generated during medical procedures, example, intubations. The vast majority of transmission is via bodily fluids. Its shelf life on fomites is short, unless it's contained within these bodily fluids, and human infection can also occur through contact with wild animals, example, bats and dogs. The main organs affected are the lungs, the liver, and the intestinal tract. This is a graph from the CDC website that shows the delayed clearance of the virus in days among the several different types of bodily fluids. So how does the incubation for Ebola work? Well, it results in an abrupt onset of symptoms between day 8 and 10 after exposure, but it can range anywhere between day 2 and day 21. The incubation period really depends on the type of exposure that you've had. For example, it takes approximately 6 days if it's been a percutaneous exposure versus approximately 10 days if it's been a contact exposure. Once inside the body, the Ebola infects these antigen-presenting cells, which are the monocytes, the macrophages, and the dendritic cells, and it also affects the neutrophils. And by activating these cells through the infectious process, a cytokine storm gets released. And these are the cytokines that lead on to produce a systemic inflammatory response that is associated with a very high mortality rate. The inflammatory response then spreads to many of the other infecting cells and organs, endothelial, hepatocytes, fibroblasts, adrenal cortical cells, and they all give rise to capillary leaks, liver failure, and shock states, respectively. Hemorrhage is usually a late-stage finding, and it occurs from the lack of clotting factors that are produced because of the hepatic dysfunction. If left untreated, death usually occurs between day 7 and 10. Ebola is a difficult virus to neutralize because of this glycan coat that it has surrounding it on the outside. It actually releases some of this glycan coat that serves as a decoy to use up many of the neutralizing antibodies that the body might have secreted in order to fight. As time goes on, this virus has the ability to diminish the body's innate immune responses. Let me explain. There are two proteins that are found within this virus, called VP24 and VP35. These two proteins downregulate the cell's protective mechanism, the interferon response, and allows massive amounts of viral titers to be produced. If you remember, interferon has several activities. One, it induces an antiviral state within the cell, making viral replication extremely difficult. Two, it upregulates the MHC molecule and promotes the recognition by the cellular immune system. And three, it supports the generation of the humoral response. What we've noticed is that if you're destined to be a survivor, it is critical to be able to mount the production of neutralizing antibodies, the humoral response. We suspect that some patients are dying before they're able to produce enough antibodies, and thus are unable to neutralize and destroy the virus. The clinical manifestations are very nonspecific. It usually has a very abrupt onset that usually occurs between day 8, 9, or 10 after exposure. day 2 through 21 to give you a bigger range. But most people fall within 8, 9, or 10 days after exposure. And like I said, initially it was very nonspecific, flu-like symptoms in nature. There's this very nonspecific erythematous macular papular rash that occurs, usually between day 5 and 6, and can desquamate. GI symptoms include watery diarrhea, nausea, vomiting, abdominal pain. There is a massive electrolyte abnormality that needs constant repletion. And there's a massive amount of hypovolemia that occurs, probably from the low serum albumin and the third spacing, usually requiring about 5 to 10 liters per day. What we've noticed is that the group that is going to get better usually start turning around between the window period of day 6 through 11. This is when you're either going to get better or you're not going to survive. And it's probably because the production of antibodies is either forming or not forming fast enough. Ebola PCR is the most sensitive and specific test available, with a turnaround time of approximately 48 hours. Without any intervention, there is a 70 to 90% mortality associated with this disease. Now, with the IV infusion and basic critical care, this number has dropped to below 50%. Our data suggests that those treated early have a high survival rate. These are a few of the current treatment regimens in the works. ZMapp is a combination of 3 different monoclonal antibodies that bind to the Ebola surface, that glycoprotein. One of the antibodies actually binds to the decoy antigens that gets released. ZMapp is made from tobacco leaves, and if given early enough, there are very good survival rate results. The problem is that it takes approximately 6 months to produce. Witnesses claim that within half an hour after administering ZMapp, the rigors, the rash, and the fever improves, and the patient is up and walking within the hour. ZMapp has not been studied in randomized controlled trials to determine whether it works and if it's safe. Survivors develop good protective antibodies, neutralizing antibodies if I may. During the convalescent phase of the Ebola infection, a patient's whole blood can be used empirically to treat with the hopes of having these antibodies neutralize the virus. BCX4430 is a synthetic adenosine analog that exhibits broad-spectrum activity not only against Ebola, but other RNA viruses. It works by shutting down the polymerase activity. It acts as an RNA chain terminator and thus does not allow the virus to continue replicating. TKM Ebola is an experimental drug. It is a combination of interfering RNAs that target Ebola proteins, specifically the polymerase, the VP24, and the VP35. It also blocks the gene that upregulates the interferon responsible for the cytokine storm. The end result is the shutting down of gene expression and thus the halting of viral replication. The U.S. Army is currently investigating this drug. AVI-7537 works against the VP24 protein, which is known to augment the interferon response. By downregulating the interferon response, it allows the patient's immune system the time to fight the virus. This drug also halts viral cell replication. Brincidofovir is currently in trials for CMV, adenovirus, and other DNA viruses. The interesting thing about this drug is that it has activity against Ebola virus, which is an RNA virus. This drug mainly works by allowing its release intracellularly, and thus increasing its activity against DNA and RNA. Vaccine design and trials are still in the works. Vaccines are designed to stimulate the immune system against Ebola virus and protect uninfected individuals who are exposed. The vaccine contain Ebola antigens. They do not contain live Ebola viruses.